Okay, we'll continue to look at how peptide bonds are formed between amino acids. Okay, so the formation of peptide bond is a condensation reaction. So therefore, there is a loss of water. The water molecule H2O would come from the carboxyl group and the amino group of the two amino acids that are going to form this peptide bond. So I circled the H2O here lah. Okay, the peptide bond is not just one line. Okay, so the peptide bond should be labeled as this CONH group. Right, this together is a peptide bond. Okay. The reverse is called hydrolysis. That's when you add a molecule of water to break the peptide bond. So you can imagine the process just reverse, right? Okay, so in this dipeptide that you see, right, after the formation of the peptide bond, there is a free amino group from the first amino acid that was not involved in the formation of the peptide bond. There's also a free carboxyl group of the second amino acid that wasn't involved in the formation of the peptide bond. Okay, so the two amino acids that form the peptide bond, right, they are now going to be called amino acid residues. Okay, so usually that's the term that we use to indicate the amino acids that are found in a peptide, whether it's a dipeptide, tripeptide, or polypeptide. Okay, so another diagram showing you the formation of 4 peptide bonds that I have circled or labeled, right, between 5 amino acids. So there's the loss of 4 molecules of water because 4 peptide bonds were formed. There's again the free amino group in the beginning and the free carboxyl group at the end. Right? So these are free groups, right? The amino groups and the carboxyl group would be labeled as the N terminus and the C terminus of a peptide chain. So in this case, you have a longer peptide chain, so we call it a polypeptide chain. The two free groups that we have mentioned, the free amino group would be the N terminus, since the amino group has an N, right? And the free carboxyl group, which has a C, would be called the C terminus. Right, so for a particular amino acid chain, right, or not amino acid chain, I mean a polypeptide chain, right? There is a directionality, right, from the N terminus to the C terminus. So if I read the sequence of this polypeptide chain, I need to start from the N terminus. Okay,